Hello everyone and welcome to Let's Talk Data. I'm Vanessa Cade and I'm with my colleague
Salvador Gesson here.
And today we're going to talk to you about statistical consultancy and what we do in that role and some of our highlights. Salvador, I was wondering if you could tell me what you do as a statistical consultant, what areas of application you work in.
Oh, well, that depends on what I'm doing, depends on all the time I've been working on different projects. But the last few years I've been working mostly on genetics, quantitative genetics, and breeding consultancy. Pretty much everything related to linear mixed models and sampling and some other strategies associated with quantitative genetics, so analyzing lots of data.
Are there particularly a species you work on?
Yes, so I work with lots of species. The ones I've been dealing most lately are sugarcane, strawberry, salmon, and then here and there comes a crazy one like shrimp, clams, some small grasses. So some more tasty than others, really. You, Vanessa, what do you work mainly on your statistical consulting?
Yes, so I work mostly in agricultural applications. So anything from animal welfare to environmental monitoring. I also work in statistical ecology and conservation type applications. Yeah.
I heard you use a lot of GenStat, is that right?
I do. Yes. So I'm also a GenStat developer. I've been doing that for about 6 years now, and I use GenStat in my day-to-day consultancy work as well. I'm both a passionate user and also a developer.
Oh, that's good. And tell us a bit about the species you have worked with. Anything tasty or anything not so tasty or?
Well, so working in agriculture, I do I've worked on lots of farmed animals. So lots of sheep, lots of dairy cows, beef cows, dairy goats, chickens, pigs, and the native animals I worked on mostly have been birds or invertebrates and the occasional skink and lizard.
Oh. So some ecology too, I guess, then.
Yes. So lots of it's to do with understanding how different pest control methods impact on an endangered species kind of outcomes, and also looking at population estimation as well.
So one question I have for you. So some of the ecological data are small databases, is that right?
Yes. Some are small and some are reasonably large. So, for example, during my PhD, I worked on a lot of bird ringing data from the UK, and that was reasonably sizable. But a lot of the data is from either small trials or small databases. Yeah. So not the scale of the genomics type data sets you work on, Salvador.
Yes, I was asking that because every year, the data sets become a bit larger. So I used to work with a breeding program that had close to 1 million records of animals. In this case was trees, so more than animals, it was trees. And then with the molecular data, we start having files that are 1 gigabyte or maybe 2 gigabytes, and it becomes a bit more difficult to explore it and read it and so on.
So how have the tools and techniques you've been using changed over time as the data sets become larger and larger?
Well, it's Originally, most of the people send me the data in Excel files, and you're probably familiar with that, for smaller
Yes.
data sets, which is not bad. It needs a bit of preparation and it needs some cleaning and so on, standardization. And that was relatively easy. I still use Excel quite a lot for sort of exploring the data. But then people started sending files in, text formats for molecular data or zip. And then reading in R for moderate size databases is okay, but then when they become really large, then you don't have enough memory. So one of the things I had to do is add more RAM memory to my laptop. That was one thing. And the other thing is, if you work with R, you realize there's different libraries to read the data. The standard functions are not very efficient, and sometimes you don't want to read all the data. Maybe you just want to filter something. So then you have other libraries, like a data table that helps you to filter and read, but it's still, they are large data sets. So
Yeah. So what's the largest data set you worked with?
I had a RISE database. I can't remember the number of individuals. It was probably 6,000 or 8,000 individuals, which was not much, but it had 2 million columns. So first, it was impossible to read it in one go, so we had to split the database into different sets. And you start working in a different way. You probably know this. You start
Yes,
working
yes.
cleaning the database by portions, if you can read the whole database. Portions in terms of columns or portions in terms of individuals. But it's the technology is getting there slowly, and there is other options. They have plenty of degrees of freedom, and that's why I was asking you about the ecological data, because one of the things it always brings to my head is when you have small data sets. I don't know, you have 20 individuals. I don't know how many lizards you had, or maybe just 100 or 80. And if you want to evaluate something, you don't seem to have much power, no?
No, so when you're doing experiments on particularly in endangered or endemic animals, the first step is always a power analysis to work out how many animals you need to give some meaningful results. And the real driver is to keep the numbers low, which brings in lots of challenges. So there's a lot of discussions you have to have initially about things you can do to reduce the random variation and to really hone in on your hypothesis and have kind of robust discussions about how people want to generalize and use the results. And the other thing that happens as well is you get an awful lot of essentially unreplicated studies. So a common example of this is if that some organisation or some government or some department, for example, is trying a new pest control regime, or perhaps they want to establish a marine sanctuary. So you'll typically have one study site, and they'll match it, maybe, or they won't match it at all, with a control site. So you actually don't have any replication of the treatments, but you have quite intensive samplings at those
The pseudo
two
replication.
sites. Yeah, it's definitely pseudo replication. So very limited in what you can conclude. You can essentially compare the sites, but you can't make any robust inference that it is to do with the treatment. You may find that the sites differ, but you hope it's to do with the treatment, but you've got no foundation to make that conclusion. So that's a common kind of challenge, I guess, with
Yes.
a lot of the statistical ecology stuff I work on.
Is the limitation of the study, and again,
yes, yeah.
you can
Yeah.
do whatever the best you can do.
Yes,
Now,
yeah.
I wonder on the same lines, do you get involved writing the manuscripts?
Yes,
Okay, good.
absolutely, yeah. So a lot of the stuff I do is contract statistical consultancy. So it depends on what the nature of that contract is. But my ideal thing is to be involved in the writing of manuscripts, or sometimes it's not a scientific manuscript that results. It may be a report for a client. So I like to be involved in that to make sure, I guess, to help ensure that the statistics is described accurately and that the
That's
conclusions
exactly what I
drawn.
was thinking. Yeah. That in the
Yes, yeah.
manuscript it says these are the limitations of the study, or these are the limitations of the inference,
And the realities of kind of practical research is you can't always do what you should do, you know, in an ideal statistical world, because there are huge resource constraints. That could be availability of sample sites, by financial constraints, and also there's a risk as well when you're doing experiments on native resources as well. You have to manage potential negative impacts too. Yeah.
well, that reminds me of studies of laboratories where you want to have the minimum number of animals or rats or rabbits. And then also I have worked in consultancy on in the medical area. And obviously you want to have the maximum power, but with the minimum number of samples. So you are always
Yes.
saying, I only want 10 patients per treatment, or 8 patients will be better. Is that going to give me the view I want to look or compare the treatments and so on? So I have a different question. When you were talking about the limitations of the study, and I thought about that, and that you have to do the best you can with your data. What do you think about transformation of the data?
So do you mean
Just using a log
transforming this?
transformation?
Oh, okay, yeah,
Because
yeah.
in ecology it's common sometimes to see like a log transformations of counts,
Yes.
or a log
Yeah.
it of proportions or something. What do you think?
Yeah, so well, the log transformation is nice in a way because it has a interpretation. It's particularly of, you know, the log transformed mean being, as opposed to the arithmetic mean being related to the geometric mean. So it's good that it has a nice way to interpret it. Other transformations aren't quite as pretty as that. The simple square root isn't, or the arcsine square root. So yeah. So how do I feel? Well, it can make analysis of it by methods which are really understandable by a wider audience, for example, and over all linear mixed models possible, which is a benefit. And also, for example, with linear mixed models, if you get into that framework, you can include rather complex correlation models, which is good, enabling you to analyze, for example, temporal data or spatial data. But you do have this difficulty, and then you've got this transformed variable, which, how do you interpret the means on that scale? How do you interpret the variances on that scale? It's
The differences
it's all different.
sometimes.
Yes, yeah. So I do transform my data sometimes. Try to avoid it if I can by, for example, if I can do a GLM with a low, certain link function, or a GLMM, or a hierarchical GLM. But sometimes it's good to be in the ANOVA or the living mix model kind of framework, and a transformation is what's required. So how about you?
Well,
do you think?
before you get into me, I asked the question because I remember in ecology, you
Yes.
seem to have people who are very strict on using GLMs or GLMMs because that's the nature of the data. It's a Poisson distribution. And some people say, no, let's just use a transformation and we can add an offset and then we'll be happy with that and use our traditional methods. So there seems to be those two schools. And the question is for you, do you consider yourself more purist or more like the pure proper distribution of the data, or more approximation and
Very good
transformation?
question. Yeah, I would consider myself a pragmatist. So I don't let things being, I guess, pure get in the way of a pragmatic solution. Yeah.
Okay, now I can answer your question.
Yes, yes.
So I think I start, and this goes back to our original discussion about consulting. It goes back to the first question, or one of the first questions you do to the client. What do you want to use this analysis for? And sometimes somebody tells you, I just want to explore. I want to see if there's something in here. Maybe we'll repeat the experiment in the future. Or it's just, sometimes they are just preliminary studies. And then you say, oh, okay, well, I can be a bit more flexible with the methods. I can use something like approximation, or I can even eventually violate some of the assumptions like independence and so on.
Yeah.
But in other cases, somebody says to you, well, no, this is for a paper that we want to possibly publish in Nature or Science. And then you think, oh, this is a bit big guns. So then in that case, I will look for the methods that seem more reasonable. And obviously, I will try to use a GLM and get away from the transformation and so on. However, I need to add that depending on the field, once you're in that area, and you probably have the same experience, sometimes I also do a bit of digging on what is accepted on the field. Because in some fields, sometimes transformations, for example, I remember in some entomology studies, transformations are relatively common, and reviewers and other authors accept them very widely. So then you can say, okay, this is the method that we should be using. Because if you use something too complicated, nobody will understand it, and then the paper might even get rejected by using the correct methods. So yes, now my consulting work is usually on companies or breeding programs. A portion of the studies gets published, but the majority is for operational decisions. So I tend to use transformations or simplifications of the models. I know sometimes I know what to simplify, but also we need to add the complexity of the data sometimes. That it makes difficult. Just a case, I remember a case where I use a generalized linear mix model for an animal model in breeding to get breeding values. And this was the heritability of maturity of fish. And the heritability was 0.92, 0.93, it was 0.9 something, which if you know the biology, you think, no, that's too high. What's going on here? This is a binary trait also, and it is very, very strange. So if I ignore the normal distribution or I use a transformation, the heritability was 0.22, for saying a number, which is more or less what you're expecting for this type of trait. So in that case, you are a bit on the decision of saying, okay, well, this is not the correct method, but it's giving me something biologically reasonable compared to a correct method that it's clearly getting lost because of the complexity of the data or something. So that's really how it goes.
Yeah, I think you brought up a really good point. In addition to the other points you've been making there, that with statistical consultancy, it's so crucial that you understand what the scientist or your client needs and how that information is going to be used. And well, in my experience, that is often easier said than done. A lot of to really understand exactly what is needed. And sometimes, in my experience, the client I work with themselves, they don't actually know what they need. They have a vague idea, and they have a vague idea of what they need, what they want, but they have a really good understanding of the challenges that they're facing in their domain or their industry, and you can be really interesting working with a client like that to devise a statistical experiment or a study that will hopefully provide them with useful information to address that challenge or problem. Yeah.
That reminds me of also the difference between working with students or graduate students, PhD students, and the faculty, because I remember cases like you want to use one method, but it's too complicated.
Yes.
And the student won't be able to defend it.
Yes, yes.
Explain it, understand it. And so, and depending on the university, depending on the team, sometimes the student can say, no, this is the method. I don't understand what's going on, but that's okay. This is what we did, etc. And sometimes they have to defend the method. So then you just have to select something that is a bit more simple to explain to. So,
yeah, absolutely. Yeah.
So, and you talk about also, I just noticed you talk about sometimes the scientists have a very good understanding of their process, but I also have the feeling that sometimes they had a very good idea of what results they want to obtain. You know, so they say, I'm sure this treatment is better because this fertilizer that I developed is better, is being better. So how do you deal with that in your experience?
So it depends on when that conversation starts. It's always easier if it starts at kind of the experimental design stage, if not earlier than that. So you can actually design a proper experiment to test the hypothesis that their fertilizer is better. If it happens after you've got a data set, you really do have to drill in and ask lots of questions, how the data arised, how they collected it, what they've done. And another issue that I've encountered is people have, say a scientist has analysed their data themselves and they haven't achieved magical statistical significance, usually less than 5% p-value. So they come to me and ask me to make it significant in the hope that I can apply a different approach. And I always feel very, very uneasy about that. And obviously I'd just apply the techniques to analyse the data that I believe are correct, and I won't go around fishing for a significant p-value. But yeah.
I've been in that situation where compare some treatments that were not significant,
Yeah.
and
Yeah.
and the scientists came back to me to say, can you try another method? And actually, I did try another method. I mean, you can sometimes go from parametric to non-parametric test, and then nothing was significant again. And then she said, oh, but maybe we can try this other method. And then we tried the other approach, and also nothing was significant. Luckily,
Yeah.
nothing was
Yes.
significant all 3 times.
Yeah.
But I was like you, we are just fishing for a
Yes.
result.
Yeah. Yeah. The other fun one I get is someone sends me data set and they ask me to find the significant results with without any context about what actually they're trying to do. And I can find significant results in any data sets if I try hard enough, but they're not going to be meaningful. Yeah.
Not for you, there'll be meaning for the scientist.
Yeah, yeah, true. Might be meaningful for anyone else though.
Yeah, I mean, we won't talk too much about this time, probably about the relationship with the scientist camps. But it's interesting that obviously we need to consider their view and their expectations, really. But also
Yes.
sometimes you have scientists that come and they treat the consultant just like I tend to think like an accountant. Can you
Mm.
please pay this invoice for me? It's almost like, can you please give me the p-value I need for this? And they go away without telling you anything about what they need, why they need it, what's the context, and so on. And don't want to go too much into details, but it feels a bit like they tend to forget that we're also scientists and we are
Yeah. Yeah.
interested on understanding the problem, not just crunching the number and so
Yeah.
on.
Yeah. If you don't have a good understanding of the problem, then you can't really be a good statistical consultant. I don't believe. You do have to be motivated and inspired by the underlying problem, and yeah, be generally interested in it. And if you are and you understand the domain, not as an expert, obviously, but you can add a lot more value. Yeah.
Fully agree. So going a bit back about your lizard study, I just bring to me the lizard. What did you do specifically there with the lizards? Do you remember
Okay.
something? Yeah.
Yes. So I've done a few on different
Wow.
lizards and geckos. So one was looking at different tracking cards. So to help estimate the population, people put out these cards with kind of ink, I guess, and the lizard walks over it and you can count how many different types of footprints you have. But there's many different ways of setting them up and different kind of lures to attract it to your tracking.
Mm-hmm.
So one thing I looked at was different methods and which one seemed to be the most accurate in terms of providing an estimate of the population. Another one was actually was helping a friend of mine who was doing her master's in Victoria University in Wellington, New Zealand. And she was interested in the rapid colour change of skinks. So she actually had a whole lot of different field trials and experiments where she tested various things related to colour change, and in particular whether it was related to a predator response. So in one of her trials, she had different models of predators kind of flying past. So different types of birds or some rats, and also she had some non-predators as well. And she wanted to see how the lizards, or skinks actually, changed
Wow.
colour in response to that. Yeah.
That sounds interesting.
Yeah. That's interesting.
But you don't have the chance to go to the field and observe some of these lizards, no?
Oh, I do, and I try and get
Oh.
out to the field to observe things as much as possible, particularly when I'm new into kind of that area, because there's nothing quite like seeing something
Yes.
to understand how the experimental survey or study is being run, and kind of the practical constraints of it. So I've done many different things. Been involved in glasshouse trials, counting tillers, for example, and that gave me a massive appreciation of the amount of work the scientists and the research technicians do in collecting this information and how valuable it actually is. Where before I was a bit almost cavalier, like, oh, you can easily increase your sample size to, 30 plants, but that'll fit in the glass house without really taking into account the amount of human resource it takes. Yeah,
It's true.
all sorts of things.
Some things it's good to go and see them and feel them a bit better.
Yeah.
And probably you also, one of the things, again, we might talk more later about is you understand more how the outliers originate.
Yes, yep. How
So
outliers originate,
an
where
unusually
things
large plant
possibly
in
go
the
wrong.
middle of the field that you think, wow, that's actually unusual, but it's there. Or something. I tend to think about trees. You see damage. So then you see like plants that were eaten by a deer or something, and then you understand. Or when people mix up, they have too many envelopes to different parts, and they might put the sample in the wrong envelope, and so on and so on.
Yes, yeah.
So, well,
Wow.
that was very good, Vanessa. I think we have a good conversation about, let's talk about data today.
Yes, we did, yeah. And we've lots of topics to talk about in more depth in future podcasts.
Exactly.
You have been listening to
Let's Talk Data. I'm Vanessa Cade.
I'm Salvador Gessen.